voice of Tom Park, who you all know. Kert Viele is going to join me. I'm Scott Berry, and just talk about the adaptive design guidance. I'll make a few comments about it. I'll pass the ball back to Kert. And Kurt's going to talk about both the guidance and the role that FACS plays in the guidance. I thought I'd make some high-level comments about it. I'll make a couple specific points about it. In general, I thought that this was absolutely wonderful, to the extent that I wish I had written it, and it's not too different from what I would have written if I had the opportunity to do that. And in many ways, why I say that is it's inviting. It absolutely talks about the key aspects of pre-specification. It talks about the need for simulation. It talks about the ability to measure operating characteristics by simulation. It justifies it as an appropriate way to calculate operating characteristics. It opens up the world that we no longer need to have an analytical solution in order to be able to run a design, but that numerical integration or simulation can be used to calculate operating characteristics, which I think opens up the door to a huge amount of adaptive designs for adequate and well-controlled trials. So I think that's great. It talks about the importance of the operational aspects to a design, which I think is valuable. A good design can go poorly if it's rated well. We thought operational bias. I think the last guidance was a little too heavy in that regard, and this one I think makes it sound like this is doable, but be careful and be smart within the trial, which I think is wonderful. So I was very pleased by it in general. I have a couple nitpicky things on it. Was pointed out by somebody else here at Berry Consultants. I agree with it, that it uses the phrase that when the design changes, and just to reinforce the idea of pre-specification, it's not the design that changes, it's adaptations that happen during the course of the design. But I think it's a bit nitpicky because the guidance is very clear on the need of pre-specification. I thought I would point out a couple interesting things within the guidance. I went through and colored various parts. The nitpicky things I thought I would talk about was, of course, it talked many times about simulations. So, for example, it says it may be necessary to use simulations to evaluate the chance of erroneous conclusions. That absolutely opens the door that that's a legitimate way to calculate probability of type 1 error, which is a large part of the document. It does talk about estimating treatment effects. I just point that out as something we haven't done that a lot. FACS is actually very good at presenting to you the bias. You can calculate for your analysis method the bias with all of the adaptive, multiple adaptive things in there. So FACS is very good at that, and it's things, but we haven't focused on that in general. So that was something that will be new for me a little bit in much of this. I will point out a couple things. I struggle with something, and this really, in a way, I don't think this is the agency's fault. And I'm not sure whose fault it is. I think it's the fact that group sequential has been so dominant within adaptive trials. But there's a notion out there that doing an interim analysis causes type 1 error inflation. And it's because almost all interim analyses that have been done in the past have been group sequential type interim analyses. And I thought there's parts to this that can cause that to be the interpretation. So adaptations based on comparative data, which essentially means unblinded, generally do directly increase the type 1 error probability and induce bias and treatment effects. I think that can be a bit misleading. Interims do not inflate type 1 error. Looking at unblinded data does not cause inflation of type 1 error. And many people believe that statement, that looking at unblinded data causes inflation and you have to pay penalties to look at unblinded data. It's the actions potentially taken at the interim that can inflate type 1 error. And then later in the document, they actually point out that response adaptive randomization can deflate type 1 error. Futility analyses deflate type 1 error. It's actually unclear whether the agency actually intends to mean that you can buy back type 1 error. I don't think so, but it actually almost opens up that door at a different point in here. So I've been trying to fight that for years. I'm a little bit worried that it opens up that door, and that continued interpretation may happen based on what they say. They're completely open to Bayesian. The choice of scale is unimportant as long as you evaluate the operating characteristics. So they're agnostic to Bayesian frequentist, which is good. I think that's relatively unimportant. I wanted to talk about one other aspect to this. Here's where it talks about response adaptive randomization alone does not generally increase the type 1 error probability, depending on the analysis technique and how that's done. Usually response adaptive randomization deflates type 1 error within the setting. And so that's the point. We have done adequate and well-controlled phase 3 trials that have been given SPAs that do upwards of 20-plus interim analyses, unblinded, where there's no adjustment to the nominal value at the end, and FDA agrees that controls type 1 error. It's typically response adaptive randomization, and there's no inflation of type 1 error due to that. And so that's a little bit of my quibble with that point. The other part is how big of a role simulations play in the document. It talks about clinical trial simulations will often be necessary to evaluate the trial design. And they go through and they do really quite a nice job of it. They talk about the simulation of type 1 error. And so this is the last part, and I'll turn it over to Kert. We have struggled with this for years and been successful, actually, at the agency. And this is the first place that they have explicitly written down that you can do this. And this is the struggle where you can walk in with a formula that shows analytical control of type 1 error and overall global null hypotheses. But in many circumstances, many good designs, quality adaptive designs, you don't have that. So here they talk about the ability to look over a grid of null hypotheses that are plausible. In the scenario, they use the word plausible for control of type 1 error. And we've done this in many settings where we've walked in over a grid of values and showed control over that grid. And of course, you can't say that every infinite possibility within that, uncountable sets of nulls are controlled, but you can show over a grid. And they don't use the term post-trial simulations, but they refer to it. And this is something we've talked about in designs, that we control over a grid, and we even specify that we will do post-trial bootstrapping to show that the actual result still was within this scenario and controlled type 1 error. And so they make reference to, in the example, if the observed placebo mortality rate was unexpectedly 50%, additional simulations would be required. And that's after the trial. So they talk about post-trial simulation. So in trial designs, we have specified how we're going to do post-trial simulations in a scenario where type 1 error is playing an important role in the trial. And so they even bring out this possibility. So I will stop there. I think Kert's going to talk about the aspect of what's submitted to the agency, the role FACS plays in that. It is a FACS webinar within the particular setting. So again, I was very, very pleased with the document, despite a couple quibbles, which I would have if I would have written it. So very, very pleased. And let me turn this back over to Kert. Tom, you may be able to. I can't see Kert.
I'm right here.
I don't believe you. Welcoming. Okay, thank you.
Yep, done it.
Welcome you to. By the way, let me throw out that to chat on the box if you'd like to everybody. I'm happy to sort of do responses, Kert as well, but Kert's going to take the ball here.
Yeah, it's a good point, Scott. Yes, yeah, do please enter questions in the chat boxes, all the lines, but Scott and Kert have been muted. But do feel free to put questions or even comments into the chat box. We'll see if one of us can notice them. But Kert is absolved from having to look at that, so he can concentrate on the slides and talking.
All right, very exciting. All right, so going to talk about the adaptive guidance first, and then get into facts. One thing that was nice about the document in particular is it laid out principles rather than a sense of you can do the following 3 designs. It made sure that these are the principles that need to be demonstrated in a design, and left a lot of opening on how you could go about that. And so that opens up the possibility of more Bayesian complex trials involving simulation. There certainly is a principle of type 1 error control, usually due to multiplicities inflating type 1 error. As Scott mentioned, that can come from multiple tests. In enrichment designs, you can see multiple groups. In dose finding, multiple doses. You do have to be careful of the type 1 error control. As Scott noted, it's not ubiquitous, that it's always inflated, but you always need to be sure and check and make sure that it is controlled. We can sometimes do this analytically, but often we do this by simulation. And when we get into the facts part of it, I'm going to show a little demo of this. Another principle is estimation, particularly the accurate estimation of treatment effects. If we get to the end of a trial and we want to say that this drug increases a response rate by, say, 18%, we want to be confident that that 18% is not horribly biased up or down due to the fact that we did an adaptive design. So the guidance makes notes of some aspects, or some designs, like group sequentials, where there are explicit bias adjustments, and leaves it a little more open in complex trials that the extent of bias in estimates should be evaluated, and notes that if there are any appropriate cautions, those should be used. So again, when I get into the facts part, I'll show some examples of how we might quantify bias and whether it's worrisome in at least one particular design. Another principle is pre-specification. This certainly, we want all the details present. So pre-specification is not, I will do a Bayesian design. It is, I will do this Bayesian design. Here are my priors. Here are my models. Here's when I'm doing interim analyses. Here's how I'm handling missing data. All of this, these aspects need to be specified in advance. And you need to do this in order to, one, just make sure that you're doing the proper analysis, but all of the operating characteristics. In order to have accurate calculations, you have to have that pre-specified. You have to have a computer program do the simulation, and to program that to make that computer program happen, you need to have all this stuff specified in advance. This also avoids uncertainties in the conduct of the trial. You're trying to anticipate what might happen so that there's no, oh, what do we do here kind of issues. I thought there was an interesting theme through the document on DMCs, talking about the potential role of the DMC in implementing the adaptive design. In a couple slides we'll talk about there's a suggestion that you could also have an adaptation committee do it. But certainly there's an emphasis on DMCs focusing on the primary responsibilities of patient safety and integrity. And so there's a lot of sense of this adaptation piece being separate from what that primary role is. It might go with the DMC, but it also might be placed elsewhere. And so part of this pre-specification is, again, to make sure the DMC is not burdened with, oh, well, what do I do in this unspecified situation? There's a lengthy discussion of trial conduct. A lot of this is related to limiting information and making sure that you've specified in advance detailed communication channels. So there's a discussion in the submission materials about making sure you've got charters and all the information about who knows what when. And so you want to restrict that information to the people who need to know. So if you're doing an adaptation, it's unblinded. There are certain people who have to receive unblinded information, but we want to make sure that that doesn't get out any more than is necessary. There's a discussion of operational bias. As Scott noted, this is similar to the prior document. There are some examples. And again, this is related to information leakage. And so the goal is to avoid the operational bias by controlling the information flow properly. There is a nice discussion of simulations. Again, the primary use of simulations in these designs is to determine the trial operating characteristics. The basic idea, of course, of simulations is pretty simple. Everything is about using the law of large numbers, and we're going to simulate a trial thousands of times, and any aspect of that trial that we want to know, we're just going to take the observed proportion. If we want to know the power, we simulate 100,000 trials. If we find that 90,000 of them were successful in a scenario, that's 90%. That's our estimate of the power. And the more simulations we do, the greater those estimates are. Those, again, just converge through the law of large numbers. There's nothing magical about this simulation idea. It's just really basic numerical integration. Use of simulations, if you can use simulation to find the operating characteristics of one design, a key strength here is the ability to compare designs. If we want to decide, are we going to do design A, design B, or design C, we certainly just want to simulate all of those and figure out which one is best. The nice thing about having a tool like FACS or an ability to do these simulations, you don't have to sit in a room and guess between A, B, and C what's better. Rather than have that meeting, somebody ought to go off and simulate designs A, B, and C, and you have a meeting to discuss the results, and instead of guessing, you've got what the right answer is. Another key aspect of this is the simulations are used to justify the design. The FDA guidance is very explicit that if you're doing an adaptive design, they want to see why. That makes complete stents. These are more difficult to review than a fixed design. They're more difficult to implement than a fixed design. The review, anyway, everything about this, there's some complexity that's added. In many cases, that complexity is well worth it, but you need to have some discussion of why you're using it. We're achieving far greater power. We're using a far lower sample size. This allows us to answer a question that we wouldn't be able to answer using a fixed design. So all of these things need to be present here, and simulations inform that. In the simulation section, Scott noted the exploration of the null space. The slide here essentially repeats that. There is an emphasis on larger simulations, particularly for type 1 error control. So they do give a number of 100,000, and that's to estimate a 2.5% rate to within 0.1%. Certainly this can be on a grid, as Scott noted. Really interesting section on information borrowing and informative priors. This is something that is available in facts. It's well known that borrowing historical information can inflate the type 1 error, and this happens in situations where there's a mismatch between the current setting and the historical setting, where you've basically had some kind of drift in, say, a control rate, for example, over time. Usually when we're using this, using historical data, we're doing it because we think that kind of mismatch is unlikely. So we think the situation where you would inflate type 1 error is unlikely to occur. But in terms of the mathematical definition, if you think of the absolute worst possible case, the type 1 error could be inflated. It's also possible it's deflated if the historical data does match. So the draft guidance has a good discussion of this, noting that maximal error as a worst case scenario. And so you end up with a discussion with the agency in that case over what that worst case scenario is, how big it can be, its likelihood, etc. And that's going to be a very subject specific discussion.
Kurt, just on that borrowing of historical data on the control, is there any opportunity for an additional justification or an alternative justification, which is that if we can't borrow historical data on the control arm, we can't do the trial, or we can't have a control arm in the trial, we will just compare against a fixed rate?
Yeah, so I think that it does depend on the setting. Particularly in rare diseases, there may be a situation where that worst case scenario, for example, may be higher in certain situations than others, and that may be motivated by simply what is possible in a scenario. This is a little bit of a sidebar, but I've always found it interesting. We often do historical borrowing in rare settings where we have limited data. In terms of assessing historical information, it almost makes sense to do this in really common settings where we have lots of historical data and we've seen the type of deviations that can occur. So I think this is an open research area, and I know there are groups working on this right now, like Transcelerate and others. There's a maintaining integrity. Again, this relates to information flow. There's a note in here that I thought was interesting. It notes that the sponsor certainly needs to know certain information. I mean, they need to know, is the trial continuing? In many cases, they have to make notifications. They have to make various disclosures about what's going on in a trial. But again, there's an emphasis on firewalling this to the bare minimum, going to the sponsor. In terms of DMCs, again, there's this emphasis on the role of safety and trial integrity. And it notes this possibility of an additional committee. We've tended to work in a couple paradigms. In one, the DMC might implement the adaptations. But for complex designs, we've often worked in a paradigm where there's an additional adaptation committee. And this appears to be endorsed by the guidance. It indicates that the structure with the additional committee may allow the expertise to be better distributed, where the adaptation committee is more focused on the statistical adaptation, and the DMC again focusing on trial integrity and safety. There's also a note here, we heartily endorse this, that those external bodies like the DMC and the adaptation committee be involved in the design stage, or at least before the trial is started. And again, this is just to limit or to gain agreement on planned adaptations and to limit surprises. The main thing that you don't want here is unplanned adaptations. So you don't want to be in a situation where the DMC sees the first interim and suddenly goes, oh, that's what you're going to do here? So you want to make sure that everybody's on board prior to the design. So this may happen earlier, it may happen at a kickoff meeting, but you want to make sure that that happens before. You want to make sure that essentially you test the airplane in a flight simulator before you actually are flying it. Again, on integrity, lots of interesting things here in this section. There's a possibility that to try to limit information leakage, notion that the protocol may only outline a general approach, which details being placed elsewhere. And so again, you may not have the exact rules in the protocol. They're certainly written down. Everything needs to be pre-specified, but this is in terms of the sites not knowing exactly what the design is, but just being given information the trial is continuing, and they know the general approach, but they may not know the details. And again, this is to limit the information to make sure people don't know, oh, an interim happened and it concluded this. So keep that information away. There's a discussion of regulatory interactions. Certainly complex designs require more review. I'm going to talk more about this when I get to the facts part in a couple slides. But again, there's a discussion of justifying the adaptive design, and also a point where it reiterates that the execution of planned adaptations, this isn't something you need to communicate to the FDA. So if an arm dropped in an arm dropping design, you just do it because that's part of the design that was the original. You're agreeing to the plan.
Kurt, sorry, there's a question being asked about, does the guidance say anything about the interaction between the DMC and an adaptation committee?
I'm trying to, so I should be careful. It's possible that it does. I do not recall that being discussed on what best practices are. It does explicitly say you need to have charters for these groups, and those charters do need to describe the interaction, but I don't think it's prescriptive on what that interaction is. Our standard model that we've followed, so this isn't explicitly in the guidance, is that the data comes from a CRO or other source. The adaptation committee prepares a report. That report is given to the DMC as either for approval in some cases, but often entirely as advisory. This is what the protocol says. We're going to do it unless you object. And it's also communicated to the sponsor. So I think that that's an issue between the DMC and the sponsor and the FDA on a case-by-case basis.
Yeah, I think that's right. I don't think they tell you what to do, but they just want to make sure that it's written down, and they refer to this data access plan that they would like to see, that the who knows what, when, and how this works is written down.
So our charters, for example, usually have very explicit discussions over what happens if the DMC disagrees with an adaptation. How do they know about it? How do they raise an issue? How is that communicated? Often that may involve a discussion between the DMC and the adaptation committee. And again, this is just getting all the relevant expertise in the same room and only involving the sponsor if the disagreement raises to a certain level. But the goal, again, is to try to, asking lots of questions is potentially an information leak. So the goal is to try to be explicit at how that was handled to minimize the leakage of information. The documentation here, there's, I'm going to skip this slide a little bit because I'm going to show you some of the things that go to the FDA, but essentially there's a lot. All the details, all the code, example trials, operating characteristics. I'm going to show you a report in a couple slides that shows all of this. We talked about charters, just did. Scott mentioned a data access plan. So again, this is all just tell us how you're going to handle this. So let me move into facts. As Scott noted, the new guidance is very welcoming of Bayesian complexity simulations. I think a lot of people reacted negatively to the less well understood language in the prior guidance, that this was discouraged. This language is gone. So it's certainly welcoming. There's explicit mention of many of the designs that are in facts. So enrichment designs, dose ranging, stage designs. It doesn't say the word Goldilocks, but designs based on predictive probabilities, which Goldilocks is. All of these are explicitly mentioned, and the document is certainly aimed at phase 3. So again, you need to justify all the principles, but these are all on the table. In terms of what we use facts for, certainly the comparison of multiple designs trying to optimize design features is our main goal. The fact that facts is C++ instead of R. R is wonderful, but R is slow. So facts is about 100 times faster. That allows you to run more simulations, allows you to do the large-scale simulations for type 1 error over a grid of possible values. It allows you to explore more designs. And certainly facts is set up so while it gives you an array of graphs and operating characteristics in the GUI, all of the output files are also available. Those can be downloaded into R for post-processing, to make graphs, to make presentations, and so forth. So let me see if I can show what that involves. So this is for type 1 error control. Sorry, somebody say something? All right. For type 1 error control, I just made a quick design up. This wasn't intended to be exhaustive, but I just want to illustrate that facts is really nice about getting you type 1 error control. This was a dichotomous example where I thought the control rate was going to be, say, around 40%. If we know dichotomous data, the type 1 error rate can change depending on how close to 50 or how far away from 50 you are. I made a really basic grid. I just did this at 30, 40, and 50. If I were doing this in a more formal setting, I might explore everything from 20 to 60, 20, 25, 30, 35, and so forth. But one thing we see here, these are big simulations. The type 1 error does seem to be a little bit inflated when I do this. If I was going for 0.05, then it's a little bit. If I were going for 0.025, then it's quite inflated. But facts, you can see, it doesn't change that much depending on the rate. So you've got that information. And facts has a graph that allows you to explore the final success criteria. And one of the nice things on here, this is a graph of the threshold versus the type 1 error rate. And you can see the threshold to achieve particular type 1 error rates. It just computes that for you. So it says I should change the threshold to 0.9812. This is a multiple arm design. There's a correction for multiplicities. If I want a new target rate, I can put that in, and you can see this automatically recalculates it to 0.9908. A best practices idea is usually to get this rate, make it a little bit lower, and resimulate under another seed to make sure you have an independent confirmation. But since the number of simulations is large, this should work well. Another aspect of this is comparing designs. So this is actually an RAR example where we wanted to determine what is, how far should we do burn-in? Should we burn in 20% of our data, 30, 40 of equal randomization before we turn RAR on? Should we do lots of interims? Should we do a few interims? Should we base our adaptation on the probability an arm is the best or just the probability an arm beats placebo? Should we, I think we had one more that was, there were 4 features here. And the last feature, we did some thresholding where we thresholded the RAR probabilities down to 0. And we wanted to vary how aggressive that was. If you put that all together, it's about 36 different designs. So that's a lot. But again, this is a matter of just running it in FACS. The FACS files look virtually identical. You can run those 36 designs. We loaded those all in to R. We wrote a little function that did the graphing. And I don't have time to explain all the details of this, but I'm hoping you will at least believe me that these graphs are informative. They say things like the power. This is what happened when we based adaptation on the probability an arm beats placebo. This is when we did it on the probability that you were finding the maximal arm. And you can see, for example, power is lower here and higher for the max. The graph's a little complicated. It also illustrates that doing many interims versus fewer was a fairly minimal difference. So if operationally it was easier to do fewer interims, we quantified the cost of this. It cost you 1 or 2% power, and that may or may not be valuable operational complexity versus the power calculation, but it's quantified here. We also found other graphs which indicated things like a 20% burn-in was optimal. And it's really, this best arm selection was a little lower, but it's pretty optimal across the board. Mean square error, we want to go down. This gets at the notion of we are quantifying estimation. FACS is producing the mean response, the variance. You can get bias, mean square error. We're reporting mean square errors here, and these were small, and in fact much smaller than you would get for a fixed design. So the RAR is being helpful here. So again, not explaining all the details of this, but just saying that FACS combined with a little graphics in R has allowed us to optimize our design, and we've got the optimal burn-in, the optimal driver of adaptation, that we know how to quantify fewer versus many interims on what the trade-off is. All of these things have been explored through the simulations, and that's gained us a fair amount. You can see in terms of power here, this is 82 versus the worst is about 78%. So that's not a trivial difference. And again, note that graph was set up to kind of focus on these upper points. There were some bad ones in here which took the power way down into the 70s. So meaningful changes in optimizing this design. In terms of pre-specification, we want to emphasize that the FACS file itself is pre-specification. Everything that you need is in that FACS file, and it's point and click. It's intended to make sure that you've thought of everything and filled in all the boxes. And in fact, that fax file, the fact that you can move it from one place to the other, and fax can be used in the design, it can be used for the submission materials, and it can be used in the execution environment. The ability to transfer that fax file assures that you have pre-specification, and in addition, to make sure that nothing is broken, because that one file can be transferred and used in all the different places. The documentation notes that you need to submit code if it's non-commercial. Fax is commercial. It is validated commercial software. It is licensed by the FDA. If you're doing supplementary things in R, you'll need to submit that, but that's certainly much less complex than your own simulation engine. And again, in terms of planning, the fact that nothing is broken, the fact you can move this file, that's ensuring the operating characteristics are being maintained. In terms of submissions, fax will generate sample reports. We have something called a report generator. Many thanks to Dr. Anna McLaughlin, who has worked tirelessly on this. Certainly those reports require some editing, but they provide a great start to you. If you have a design in fax, you can get a start on this, and then it needs to be cleaned. At Berry, in terms of our infrastructure in doing this, we automate our report generation using SWeave. This is standard functionality in R. This essentially allows you to have generate a Word document or a LaTeX document, but it allows you to have all the text, and then everywhere that there's a number that needs to be filled in, what SWeave allows it to do is it allows R to compute what that number is, or more likely, because we have the fax output, R can read in the fax output and then put those numbers in the report. And so this is nice. Kind of the worst thing that happened, not the worst thing, but a bad thing that happens is you've done a whole report, somebody wants to change something, and now it's, oh, please don't make me rewrite the whole report or do a lot of cutting and pasting to edit it. And so the fact that this is all automated in SWeave, if you wanted to do something like, oh, our accrual rate, we thought it would be 10 per month, but now we think it might only be 6. What does that do? Well, you change the 10 to 6 in fax, you rerun, you get the output, you rerun the SWeave code, and there is your report with all of the operating characteristics for an accrual of 6. And this is something that you can do in minutes rather than taking a long time trying to get all this redone. A sample report, let me find that, here we go. This is a very generic, so you can see it's a confirmatory study of some drug and some indication. This is not a real example, but it's inspired by one. Again, introductions on what the trial is doing. Part of the submission materials is everything has to be explicit. So a discussion of arms, endpoints, the dose response models that we're using, the priors that are involved in this. These all need to be written out. Some justification of how these are non-informative, why we're using those priors. Discussion of how we're computing posterior distributions. A discussion of the adaptive design. When are we doing the interim analyses? How are we allocating patients? What are our criteria for stopping accrual? And again, these thresholds all need to be placed in here. At the end of the trial, what hypotheses are our testing? What's our final analysis? How do we know whether or not we've won or not? This one has a schematic on how things work out. The FDA is explicit that example trials are valuable. These are often situation dependent. So I don't have movies in this particular document, but our adaptive design reports, they all contain 1, 2, 3 or more example trials that go through, here's what happened at each interim, here are the model fits, here are the decisions that were made, and here's why those decisions happened. Again, it provides something concrete so you can illustrate all the rules and get an idea of the face validity of the trial. Details of the simulations. What kinds of scenarios did we do the simulations in? We have a null, for example. This was a continuous example, so there isn't as much dependence on the null rate, but there is dependence on the accrual rate here. So a discussion of simulating multiple rates. In this generic report, I haven't filled all this in, but it would be filled in. This does have a graph that shows the type 1 error rate as a function of accrual. And so this is getting at the notion of exploring on a grid. What you can see here is the accrual is as long as the accrual is about 1.5 or greater, say 1.5 greater patients per month or per week, then it looks like type 1 error is controlled. Certainly you would interpolate this as a smooth function, but if the accrual rate goes down below 1, then the type 1 error starts to inflate. And so this is the situation Scott was talking about post-simulation. If they were to accrue really, really slow, we might need to do post-simulation and go, this isn't the right threshold anymore. This needs to be brought down. More discussion of type 1, operating characteristics, power. Again, the goal of here is not to say with everything that's in this report, but just to emphasize the thoroughness, and all of these numbers are coming from the fax output. What we're using R for is to get this typed in and get this all formatted well. And again, once this automated structure exists, if you change the design, you can change the report. So let me move back. Am I doing good on time? In execution, fax is also, we talked about the value of taking that fax file from the initial design and being able to use it directly. You can call fax from R. So last week, for example, we did an analysis of a particular trial, and what we were able to do is put in, this was a sensitivity analysis. The sensitivity analysis was in fax. We took the fax file and were able to run the sensitivities just by calling the fax analysis from R. As with the adaptive design reports, we automate these with SWeave. And again, the goal is fast execution. The goal is to have all this set up so that once the data is delivered from the CRO or other source, this goes into fax. Fax produces output, and then that output is processed by R and SWeave, and then this has to be reported to the monitoring committees as dictated in the communication plans. So this is what we discussed about this adaptation committee, the DMC, all of this communication has to be laid out. The kind of reports that are generated here, let me get to this one. Again, this is entirely generic, but again, a discussion of the trial. This is the information that would go to the monitoring committee or be prepared by the I should be careful. This is what would be prepared by the adaptation committee and could be reviewed by the DMC as appropriate. Discussion of the trial, discussion of the current status. So it talks about this is the second interim. Here are some of the key things that decide whether the trial continues or not. And so you could see possible options are to stop the trial for anticipated success or futility. This is a situation that we're continuing to the next analysis because our threshold is 0.95. We're only at 0.759. It wasn't low. We're not futile. It gives a discussion of where all the subjects are. So here is all the data. Reminders of what the primary endpoint is. More discussion of the data and further breakdown. The model fit. So here are the priors. Here is what the model says is happening on standard of care and this generic treatment, some graphs of the posterior. Again, all of these are going to be subject specific, but I just want to emphasize all of this can be done automatically, and the information to make all these graphs and fill in all these numbers, that's coming from the fax output, and we're using R to do the processing of this.
Kurt, the FDA also,
yes.
they also talk in the guidance about submitting material after the fact of what happened at the interims. This becomes a very nice thing to submit exactly what happened at the interim.
Yeah, so this is actually a really important point. If you're doing these, one role of that adaptation committee is to archive all of this material, all of the data that was used. If we're doing this in fax, we would archive, here is the data file, here is the fax executable we used, here is the parameter file, these were the output files, and then here was the SWeave file that we used and the report. All of that would be sitting archived, and when the trial is done, that would be delivered to the sponsor, and that would become part of the submission. Let's see, I think this is just more graphs just to indicate there may be a lot of stuff going on. You have lots of predictive probabilities. There's some justification of this. We find that this report is something that's often done in collaboration. We prepare an initial version, and then with discussion with the adaptation committee, the DMC, the sponsor, this may evolve. The sponsor doesn't get to see them as they're being run, only at the end, but the sponsor certainly has input in what these reports contain, knowing that they're eventually going to be consuming them. I think that's all of that. I've tried to leave time for questions, so let me just put a summary here. Certainly the new guidance is welcoming of simulation and complexity, and one thing we're hoping, this is a fax webinar, we're hoping fax provides a key tool and can be part of this full infrastructure for planning, submission, and execution to make all of these things happen within a validated environment that you can use for all of these different pieces. So let me close there, and we're certainly available to take questions.
So I don't have any further questions on the chat at the moment, Kurt. I'll give people a moment to type something in. We had one question during the talk from Bill Brooker, but I think Scott has satisfactorily answered that with his chat answers. So I had just a couple of thoughts which we can buy time for people to enter questions with. One was, how well does fax do for this simulation over a grid of the null? Does it, can you actually get all the null cases you want into kind of one fax run?
So in a couple ways, certainly exploring like dichotomous rates, which are a common aspect of the grid. That's just multiple scenarios. That's quite easy. Simulating multiple accrual profiles is another one. That's easy within fax. I mean, you can end up with a lot of things in your grid. We often pick a default. You know, here's our base accrual and our base control rate, and we do one-offs. So we take our base accrual with varying rates, and we take our base rate with varying accrual to simplify it a little bit. I think if you were talking about a baseline hazard, you could also simulate baseline hazard rates. So I think you're in good shape. I'm sure someone will think of something I didn't think of, but certainly the things we've encountered in practice, fax does well. Scott?
I'll agree with that. In part, what fax doesn't do well is if you have to go over different designs because it's a different fax file. And that can be harder as the statistician to simulate them. But because you have the final design, you can create 50 nulls and in one fax file and run. So it's actually really quite good at that.
And then there's a question about, I mean, one of the, fax helps you with calculating the bias, but it doesn't actually have the an output which is the explicit, this was the bias. When you're sort of trying to estimate the bias from simulations, how conditional should that be? Do you just want the bias in the estimate of the treatment rate for each arm over all the simulations, or do you want it dependent on whether the trial was successful, whether this was the arm that was selected, and that sort of thing?
Scott?
Oh, okay, sorry, I was mute button. So it's I think you want to do it unconditional. I mean, the definition of bias is from the beginning, if you run the trial, here's the estimate that comes from it. And part of the problem, and this is what the group at McMaster did, is if you condition on successful trials, that's a bias. So it can be confused as to the adaptation creates the bias, when in fact it's the successful trials. So yes, in general, I think you want to simulate 1,000 trials under a scenario and look at every single trial at the outcome of that, and not condition on certain things happening, because that condition can create the bias, not the design. The hard part about bias is it depends on the scenario. If you run the null, there may be no bias, but if you run a case where it's successful, there may be a negative bias. There may be a positive bias in the null and a negative bias in an alternative. So we tend to report it by scenario, though it tends to be something we haven't really focused on a lot.
One last question just going back to the simulation for the nulls over the grid. Something that FACS does by default is to use the same random number seed for each scenario. So I'm just a little worried that might give you kind of excessive correlation in the simulation results. Is that a problem? Should we have a feature? Should we have an option to change the random number seed per scenario?
So that's an interesting question. Certainly, our best practices. I'm gonna answer a different question first, but I will come back. We usually determine thresholds for type 1 error control with one set of simulations, and then change the seed in order to confirm them. And so certainly, I think that's valuable. In terms of having multiple seeds for each element in the grid, I think that certainly makes sense. If the simulations are large, I don't know that it makes a huge difference in practice. So there's certainly this aspect of I think you ought to do it, but I'm not sure that I lose a lot of sleep if it's not done, as long as the simulations are big.
No, thank you very much, Kurt. Well, we're at the top of the hour, and we have no further questions coming in on the chat. So I think we can draw the proceedings to a close. Thank you very much, Kurt and Scott.
Thanks, Tom. Thanks for doing that, Kurt. Thanks, everybody.